Welcome to another deep dive. Today we're digging into a really foundational text meant, genetics. Analysis and Principles, the 7th edition by Robert Brooker. We're going to pull out the key ideas on population genetics for you. We also think about genetics in terms of individuals like your genes, your family, but what happens when you zoom out? Look at the whole group. That's what we're doing today, exploring how genes operate and, crucially, change within entire populations over time. It's a different perspective.
It absolutely is. Population genetics really shifts the focus. We're moving beyond sort of the static blueprint of one person to this dynamic, evolving genetic landscape of a whole group. It's about the genetic makeup of the crowd,
Right.
how it gets measured, and the forces that reshape it generation after generation.
Okay, so let's nail down some basics first. What exactly is a population in this context?
Right, so a population is basically a group of individuals, same species, living in the same area, and importantly, they can interbreed with each other.
Seems simple enough.
Yeah, but within that, you often have what we call local populations. Think about the large ground finch on Daphne major in the Galapagos. They mostly stick to that island and breed among themselves, a local group.
But they could potentially fly elsewhere.
Occasionally, yeah, a bird might make it to another island. So there's this possibility, even if it's rare, for genes to flow between these local spots. It hints at the connections.
Okay. And if the population is the group, what's the gene pool?
The gene pool is essentially, well, all the alleles for every single gene within that whole population. It's like the complete genetic library they have to draw from for the next generation.
Got it. And when you look at these gene pools, you see variation, right? Lots of it.
Exactly. That visible variety is what we call polymorphism. Literally many forms. Think of the Hawaiian happy face spider, same species, but wow, they have all these incredible color and pattern differences. That's polymorphism in action.
And what's causing that at the DNA level?
Well, fundamentally, it's down to tiny differences in the DNA sequence. The most common culprit is the single nucleotide polymorphism, or SNP. It's just a single letter change in the DNA code.
These are common.
Unbelievably common. SNPs actually account for something like 90% of all DNA variation between humans. Turns out your average human gene, maybe a couple thousand base pairs long, probably has around 10 different polymorphic sites. So variation isn't unusual, it's pretty much the norm. The classic example is the beta globin gene, where a single SNP gives rise to the HbA and HbS alleles, one causing sickle cell anemia.
Okay, so tons of variation. How do we actually measure it, put numbers on it?
That's where allele frequencies and genotype frequencies are essential. They're our core metrics.
Can you give us an example?
Sure. Imagine 100 frogs, let's say 64 are dark green, GG, 32 are medium green, GG, and 4 are light green.
Yep.
To find the frequency of the G allele, we count up all the Gs. There's 1 in each heterozygote, so 32, plus 2 in each light green homozygote. So 2 times 4 is 8. That's 40 G alleles.
Okay, 40 Gs.
And the total number of alleles in the population is 202 alleles per frog times 100 frogs. So the frequency of G is 40 divided by 200, which is 0.2 or 20%.
And the genotype frequency for, say, GG?
Simpler. That's just the number of G frogs divided by the total number of frogs. So 4 out of 100, which is 0.04 or 4%. These numbers give us a precise snapshot of that population's genetic makeup right now.
This feels like it's leading somewhere mathematical.
It is. It leads us straight to the Hardy-Weinberg equation. P squared plus 2pq plus q squared equals 1. Developed way back in 1908 by Godfrey Hardy and Wilhelm Weinberg independently. Simple equation, huge implications.
So P and Q are the allele frequencies.
Exactly. P for the dominant allele frequency, Q for the recessive.
Yeah.
And the equation basically predicts what the genotype frequencies should be if the population is in Hardy-Weinberg equilibrium, meaning if it's not evolving for that specific gene.
This is like a baseline, a null hypothesis.
Precisely. It's our evolutionary yardstick. If a population's actual frequencies don't match the Hardy-Weinberg prediction, that's a huge clue. It tells us that one or more evolutionary forces are acting on that population, causing change. It's a tool for detecting evolution in action.
But for this equilibrium to hold, certain conditions have to be met, right? Like a lot of them.
Oh, yeah. 5 very strict conditions. No new mutations happening. The population has to be massive, infinitely large technically, so random chance or genetic drift is negligible. No migration, no individuals coming in or leaving. No natural selection, meaning all genotypes survive and reproduce equally well. And finally, mating has to be completely random.
which basically never happens in the real world.
Pretty much never perfectly, and that's the point. Because real populations deviate from these conditions, they evolve. The Hardy-Weinberg equation highlights those deviations for us.
So how do we use it?
Well, we saw with the frogs, if p 0.8 and q.22, the equation predicts 64% G, 32% G, 4% G. If our observed match, it's in equilibrium. But we can also test real data. There was a study on the MN blood group in an Inuit population. They counted the MN, and NN individuals, calculated the allele frequencies, plugged them into Hardy-Weinberg, and then used a statistical test, a chi-square test, to see if the observed numbers matched the expected.
And did they?
In that case, yes, they did. The population was in Hardy-Weinberg equilibrium for the MN gene, suggesting none of those evolutionary forces were significantly altering frequencies at that time.
Okay, so it's a powerful tool. What does it mean for, human health?
Hugely practical application. Estimating carrier frequencies for recessive diseases. Take cystic fibrosis. In some northern European populations, about 1 in 2,500 people have the disease. They're homozygous recessive, let's say A. So the frequency of the genotype Q is 12,500. If Q is called 500, then Q, the frequency of the A allele, is the square root, which is 150, or 0.02.
Right.
And since P plus Q equals 1, the frequency of the dominant allele AP must be 1.02 equals 0.98. Now we can calculate the frequency of heterozygous carriers, AA, which is 2PQ. So 2 times 0.98 times 0.02. That comes out to about 0.0392, or nearly 4%.
Wow. So even though only 1 in 2,500 has the disease, almost 4% of the population carries the allele.
Exactly. This equation lets us see that hidden genetic variation within the population. It's incredibly useful.
So all these potential changes, these deviations from equilibrium, fall under the term microevolution.
That's the one. Microevolution is just defined as changes in a population's gene pool, those allele and genotype frequencies from one generation to the next.
And you mentioned mutations earlier. They introduce new variation, but they're not the main drivers of frequency change.
Right. Mutations are the ultimate source of new alleles, the raw material, but they happen really slowly, like maybe 1 mutation per million gene copies per generation. So on their own, they don't drastically shift frequencies quickly. They provide the sparks, but other things fan the flames.
And those other things, the main engines.
Yeah. The big 4 evolutionary mechanisms: natural selection, genetic drift, migration, and non-random mating. These are what really drive significant changes in allele frequencies.
Let's tackle the big one first.
Yeah.
Natural selection. Darwin and Wallace's idea.
Yep. And the key concept here is Darwinian fitness, which isn't about being the strongest or fastest necessarily. It's purely about reproductive success. How likely is a particular genotype to survive and reproduce, passing its genes to the next generation?
How do we measure that?
We use relative fitness, symbolized as W. We assign the most reproductively successful genotype a fitness of 1.0. Then we measure the success of other genotypes relative to that. So if genotype AA has W 1.0 and AA only produces 80% as many offspring on average, its relative fitness is W 0.8. Maybe genotype only manages 20%, so it's W 0.2. Fitness can involve survival, finding a mate, fertility, anything affecting reproductive output.
Okay. And selection doesn't always work the same way, does it? There are different patterns.
Exactly. We usually talk about 4 main patterns. First is directional selection. This is probably what most people think of selection favors individuals at one extreme end of the trait distribution.
Like pushing the population in one direction.
Precisely. This happens when, say, a new beneficial allele pops up, or the environment changes, making one extreme phenotype suddenly advantageous.
Got an example?
Oh, definitely. Insecticide resistance is a classic. There was an experiment with mosquitoes, Aedes aegypti. They exposed lauren to DDT, and after just 7 generations, nearly the whole population was resistant. That's rapid directional selection.
Wow, only 7 generations.
And then there's the amazing work by Peter and Rosemary Grant on the Galapagos finches. They studied medium ground finches on Daphne major for decades. In 1977, there was a severe drought.
I remember reading about this.
Yeah. The drought wiped out the plants that made smaller, softer seeds. Only plants producing larger, tougher seeds survived. Suddenly, finches with deeper, stronger beaks had a huge advantage because they could actually crack those tough seeds.
So birds with smaller beaks struggled.
They starved, many of them. The larger-beaked birds survived better and reproduced. The result: in just 1 generation, the average beak depth in the offspring population jumped from about 8.8 millimeters to 9.8 millimeters. A textbook case of directional selection driven by environmental change.
Incredible. Okay, what's the next pattern?
Balancing selection. This is interesting because it actually maintains genetic diversity. It keeps two or more alleles present in the population over time. It challenges the idea that selection always eliminates the less fit alleles.
How does that work?
One major way is through heterozygote advantage. The absolute classic example is sickle cell anemia and malaria.
Oh, right.
Yeah. If you have two copies of the sickle cell allele, HBS, you get severe sickle cell disease.
Mm-hmm.
If you have two normal alleles, HBA, you're fine for blood cells but highly susceptible to malaria. But if you're heterozygous, HBS,
You get the advantage.
exactly. You might have mild sickle cell trait, but you have significant protection against malaria, like 10–15% higher survival rates in malaria-prone regions. So where malaria is common, selection actively favors the heterozygotes, which keeps both the HBA and HBS alleles circulating in the population, even though HBS is harmful when homozygous.
Fascinating trade-off. Any other ways balancing selection happens?
Another cool one is negative frequency-dependent selection. Here, a genotype's fitness actually decreases the more common it becomes.
So being rare is an advantage.
Sometimes, yes. Think about the elderflower orchid. It comes in yellow and purple varieties, but neither produces nectar. Pollinators, like bumblebees, might learn to avoid the most common color because they keep visiting it and getting no reward. This means they then tend to visit the rarer color more often, giving it a temporary reproductive advantage. This switching behavior helps maintain both colors in the population.
Clever. Okay, third pattern.
Disruptive selection, sometimes called diversifying selection. This one favors two or more different phenotypes and selects against the intermediates.
So it pushes the population towards multiple distinct forms.
Right. It often happens in patchy or heterogeneous environments where different forms are suited to different niches. A great example is the land snail, Sepia nemoralis. They have shells with different colors and banding patterns.
I think I've seen pictures. They're quite variable.
Very. And researchers found that their predators, thrushes, hunt by sight. In dark, woody areas, snails with plain brown shells were better camouflaged and survived better. In grassy areas with patches of sun, yellow banded shells were harder to spot. In leaf litter, pinkish shells did well. So selection favored different forms and different microhabitats within the same overall area, maintaining that diversity.
Makes sense. And the last one.
Stabilizing selection. This is kind of the opposite of disruptive. It favors the intermediate phenotypes and selects against the extremes.
So it tightens the distribution around the average.
Exactly. It tends to reduce genetic variation for that trait. A classic hypothesis relates to clutch size in birds, proposed by David Lack. Laying too few eggs means low contribution to the next generation. But laying too many eggs might mean the parents can't feed all the chicks adequately, or the chicks are smaller and weaker, reducing overall survival.
So there's an optimal middle ground.
Right. Selection favors birds that lay an intermediate number of eggs, leading to the highest number of surviving offspring. We see this pattern in species like the collared flycatcher. Stabilizing selection keeps the clutch size clustered around that optimum.
Okay, selection makes sense. It's about fitness, adaptation. What about genetic drift? You mentioned that earlier.
Genetic drift. This is all about random chance. It refers to random fluctuations in allele frequencies from one generation to the next, simply due to sampling effects which individuals happen to reproduce, which alleles happen to get passed on.
So it's not about being better adapted, just lucky.
Yeah, pretty much. And its effects are much, much stronger in small populations. Think about flipping a coin. If you flip it 1,000 times, you expect roughly 500 heads and 500 tails. But if you only flip it 10 times, getting 7 heads and 3 tails wouldn't be that surprising.
Yeah, random chance has a bigger impact on small samples.
Exactly. Same with alleles. In a huge population, random events tend to average out. But in a small population, an allele's frequency can bounce around dramatically just by chance. Over time, drift often leads to an allele either being lost entirely, frequency hits 0, or becoming fixed, frequency hits 100%, purely randomly.
And you mentioned how population size matters for how fast this happens.
Absolutely. For a brand new allele in a population of, say, just 20 individuals, the probability it's lost just by chance is huge, like 97.5%, even if it's slightly beneficial. Fixation also happens much faster in small populations. In a large population, say 1 million individuals, drift still happens, but it takes an incredibly long time, maybe millions of generations, for an allele to become fixed just by chance.
Are there specific situations where drift becomes really important?
Definitely. 2 classic scenarios highlight its power. First is the bottleneck effect. This happens when a population suddenly crashes in size due to some random event, a flood, a fire, disease, massive habitat destruction.
Wiping out most individuals randomly.
Right. The few survivors are essentially a random sample of the original gene pool. Their allele frequencies might be very different just by chance, and they've lost a lot of the original genetic diversity. Even if the population size recovers later, that genetic signature of the bottleneck often remains. The African cheetah is a prime example. They have incredibly low genetic diversity, thought to be the result of a severe bottleneck maybe 10, 12,000 years ago.
And the second scenario.
The founder effect. This occurs when a small number of individuals break off from a larger population to start a new colony somewhere else.
Like pioneers.
Exactly. That small founding group carries only a fraction of the original population's genetic diversity, and the allele frequencies in the founders might differ significantly just by chance. A famous human example is the Old Order Amish of Lancaster County, Pennsylvania. They descended from just a few founding couples who emigrated in the 18th century.
And they have some specific genetic traits.
They have a remarkably high frequency, about 7% of Ellis-Van Creveld syndrome, a rare form of dwarfism caused by a recessive allele. This high frequency isn't because the allele is advantageous, it's because, purely by chance, one of the original founders happened to carry that rare allele, and it became amplified in the subsequent generations due to the small founding population size. That's the founder effect.
Okay, drift is randomness. What about migration?
Migration, or more accurately, gene flow, is the movement of alleles between populations. When individuals move from one population to another and reproduce, they carry their alleles with them, changing the allele frequencies in the population they join.
So it mixes things up.
It does. Imagine you have population A where allele X has a frequency of 0.7 and population B where it's only 0.3. If a bunch of individuals migrate from A to B,
The frequency of X in population B will go up.
precisely. A simple calculation shows even a moderate number of migrants can shift frequencies noticeably in just one generation. Gene flow has two main effects. It tends to reduce genetic differences between populations, making them more similar over time. And it can increase genetic diversity within a population by introducing new alleles that might have originated elsewhere.
So it acts as a homogenizing force between populations, but can boost diversity within them. Interesting.
It's a key connector in the genetic landscape.
All right, the last of the big four. Non-random mating. You said random mating was a Hardy-Weinberg assumption, but it's often violated.
Very often. Animals, including humans, often choose mates based on certain characteristics. This is non-random mating. One type is assortative mating. Positive assortative mating is when individuals choose mates with similar phenotypes to themselves. Negative is when they choose mates with dissimilar phenotypes.
And then there's inbreeding.
Right. Inbreeding is mating between genetically related individuals, cousins, for example. The opposite is outbreeding, mating between unrelated individuals. Now, the crucial point here is that non-random mating, including inbreeding, does not change allele frequencies directly.
Wait, really? It doesn't change allele frequencies?
Nope. Think about it. If individuals just choose mates differently but still pass on the same alleles overall, the allele frequencies in the gene pool don't change. What does change significantly are the genotype frequencies.
How so?
Inbreeding specifically increases the proportion of homozygotes and decreases the proportion of heterozygotes compared to what Hardy-Weinberg would predict.
Why does that happen?
Because related individuals are more likely to share the same alleles inherited from common ancestors. We can quantify this using the inbreeding coefficient F. It represents the probability that two alleles for a given gene in an individual are identical by descent, meaning they are both copies inherited from the same ancestor.
Can you calculate that?
You can, by tracing pedigrees. For example, the child of first cousins has an inbreeding coefficient F of about 0.0625, or 6.25%. This means for any given gene, there's a 6.25% chance they inherited two copies of the exact same ancestral allele.
So more homozygotes, and that can be bad.
It often is. This leads to inbreeding depression. Because inbreeding increases homozygosity, it also increases the chances of an individual having two copies of a harmful recessive allele. Many populations carry these detrimental alleles at low frequencies, usually masked in heterozygotes. Inbreeding brings them out into the open in homozygous form, leading to reduced survival, fertility, or overall fitness.
Is this a real problem for wildlife?
A huge problem, especially for endangered species with small populations. The Florida panther is a tragic example. Severe inbreeding led to problems like poor sperm quality, heart defects, and susceptibility to diseases. Conservation efforts actually involve introducing panthers from Texas to increase genetic diversity and alleviate the inbreeding depression. It shows how population genetics directly informs conservation strategy.
Wow. Okay, we've covered the forces that change allele frequencies. But let's circle back to where variation originates. You said mutations are the ultimate source, but are there other ways novelty arises?
Absolutely. Beyond those slow and steady point mutations, sexual reproduction itself is a huge generator of new combinations of alleles through independent assortment and crossing over during meiosis. You also get things like interspecies hybridization, creating new mixes, and in prokaryotes, they have mechanisms like conjugation, transduction, transformation, basically ways of swapping DNA directly.
But you emphasize mutation rates are low.
They are. A mutation rate of, say, 10 to 5 only shifts an allele frequency very slightly over many generations. Again, crucial for providing the raw material, but not for rapid frequency changes.
Are there more dramatic ways new genes or gene functions can arise?
Yes, definitely. One fascinating mechanism in eukaryotes is exon shuffling. Exons are the coding parts of genes. Sometimes an exon, along with its flanking non-coding introns, can get copied or moved from one gene and inserted into a completely different gene.
Like snapping together Lego blocks from different sets.
Kind of.
Yeah.
This creates a new hybrid gene encoding a protein with an extra domain, potentially leading to a totally novel function. It's a way to build genetic complexity by recombining existing functional units.
Cool. What else?
Another big one, especially in the microbial world, but also impacting eukaryotes, is horizontal gene transfer, or HGT. This is when an organism gets genetic material directly from another organism that isn't its parent.
Like borrowing genes.
Exactly. A classic scenario is a bacterium getting engulfed by a eukaryotic cell, and somehow one of the bacterial genes gets transferred and integrated into the eukaryote's own chromosomes. It's surprisingly common, especially among bacteria. Estimates suggest maybe 20–30% of the genetic variation in some bacterial species came from HGT. Think about antibiotic resistance genes spreading rapidly between different bacterial species. That's often HGT at work.
That's a major evolutionary shortcut.
It really is. And then there's variation arising from changes in repetitive sequences. Our genomes are full of short DNA sequences repeated over and over again in tandem.
Like satellite DNA.
Yeah, things like microsatellites, very short repeats, just 1 to 6 base pairs like CA repeated many times, and mini satellites with longer repeat units. These regions are mutation hotspots, particularly prone to errors during DNA replication where the polymerase can kind of slip, adding or deleting repeat units.
So the number of repeats varies a lot between individuals.
Yeah, hugely. And this variation is the basis for a very practical technology, DNA fingerprinting or DNA profiling.
Ah, the crime shows.
Exactly, because the number of repeats at multiple microsatellite locations is highly variable between unrelated individuals. Analyzing these patterns creates a unique genetic profile, like a fingerprint.
So main uses are forensics.
Forensics is a huge one, matching DNA from a crime scene to a suspect. But also relationship testing, like paternity tests. Because you inherit your microsatellites from your parents, close relatives share much more similar patterns than unrelated individuals. An offspring will share roughly half of their bands or peaks with their mother and half with their father. It directly uses that population-level variation for individual identification.
So we've gone from the whole population gene pool down to these unique individual patterns based on repetitive DNA. That's quite a journey.
It really covers the spectrum of how genetics operates at the population level.
Well, this has been a fantastic deep dive. We've gone from defining populations and gene pools, measuring variation with allele frequencies, using Hardy-Weinberg as our evolutionary baseline, and then exploring the major forces, selection, drift, migration, mating patterns that drive change. Plus, we looked at where all that variation comes from in the first place. You, our listeners, should now have a much richer understanding of this dynamic field.
Absolutely. And maybe a final thought to leave you with. Consider how these mechanisms we've discussed, the subtle pressures of selection, the random jolts of drift, the connections forged by migration, even the choices involved in mating, plus the constant bubbling up of new variation, how these aren't just abstract concepts from a textbook. They are ongoing, active forces shaping the genetic destiny of literally every species on Earth, including us right now. As you look around, what patterns in nature or even human society might reflect these powerful genetic forces at play? What new questions does this spark for you?
A great question to ponder. Thank you so much for joining us on the deep dive, and thank you, our listener, for being part of the Last Minute Lecture family.